The DHA fraction of omega-3 fatty acids has been shown to provide a wide range of benefits for infant and adult brains. In addition to improving cognitive performance, research has found that DHA can play an important role in reducing the symptoms of depression, anxiety disorders, and anger. One of the major building blocks in the brain, the omega-3 fatty acid DHA, is critical for the optimal brain health and the function at all ages of life. Researchers are now finding DHA provides brain-boosting benefits in infants and in aging adults. Recognizing DHA's crucial role in neurological and visual development in infants, manufacturers are rushing to incorporate DHA into infant formulas and baby food. Similarly, adults seeking to stave off psychiatric and neurological ailments like depression, post-traumatic stress disorder, and Alzheimer's disease are now paying careful attention to their DHA intake. The omega-3 fatty acid DHA is crucial for the healthy structure and the function of the brain. An optimal intake of DHA is especially essential for pregnant and nursing mothers to ensure adequate brain development in their children. DHA is essential for the adult brain, where it impacts the brain structure and signaling systems. It helps to promote the nervous system development and the optimal memory function. A DHA deficiency has been linked with many psychiatric disorders like depression, suicidal behavior, anger, and hostility. Evidence suggests that adequate DHA intake can help to prevent age-related memory decline and Alzheimer's disease. Most scientists and medical authorities agree that a higher intake of omega-3 is important for good health. This is based on a large number of well-conducted studies in humans and animals, which have shown that omega-3 oil improves brain function, decreases inflammation, reduces the incidence of fatal heart attacks and ischemic strokes, improves the outcome of autoimmune disease, and improves vision. Of particular interest are the beneficial effects of omega-3 oils on neurological function and brain protection. For instance, a number of studies have shown that a higher intake of omega-3 oils significantly reduced the incidence of Alzheimer's disease, as well as vascular dementia, and improved the quality of life and memory in those who are affected by dementia. Although there's a general agreement about the health benefits of omega-3, few appreciate the differences in their various components called the EPA and DHA fatty acids. These two components make up the bulk of the omega-3 oils. The ratio of the EPA and DHA can vary considerably in commercial products, however, most of which have much higher concentration of EPA, mainly because it's less expensive to manufacture and has a less fishy odor than DHA. Yet studies have shown that most of the neurological benefits from omega-3 are derived from the DHA component and not the EPA. In both the Chicago and Rotterdam studies, researchers found a 60% reduction in the incidence of Alzheimer's in those who were eating at least one fish-containing meal per week. In the Chicago study, DHA exerted a strong protective effect, and this was not seen in EPA. Furthermore, the study found the intake of plant-derived omega-3 fatty acids, alpha-linoleic acid, was associated with a reduction in the risk of Alzheimer's disease in subjects carrying the ApoE4 gene, which is a powerful indicator of the disease. People with Alzheimer's disease have dramatically lower levels of DHA in the neurons of their hippocampus, an area of the brain that's severely affected in the disease. This area is vital for creating recent memory, or so-called working memory. Prasad and co-workers from the University of Kentucky found the phospholipids, like phosphatidylethanolamine, which normally contains the highest level of DHA, is severely depleted in those regions of the brain most affected by Alzheimer's disease. DHA not only improves the memory in cases of Alzheimer's, but it's also been shown to improve age-related memory loss. Animal studies have shown DHA supplements can counteract learning difficulties when soluble amyloid beta, the most injurious form of Alzheimer's associated amyloid plaque, is infused into the animal's brain. Animal studies, both in mice and non-human primates, also show DHA-depleted diets impair learning and memory, and that re-feeding DHA-containing diets reverses these impairments. Feeding rats diets high in DHA improves working memory, very short-term memory, and reference memory, or longer term, in both old and young animals. Oregon Health Science University found rhesus monkeys who were fed a long-term omega-3 deficient diet began to show stereotype behaviors during early life. These are the type of repetitive behaviors seen with social deprivation and autism. Clearly, omega-3 oils like DHA play an essential role in learning, memory, and behavior. A growing number of studies in both human infants and animals are showing a strong correlation between the intake of omega-3 oils, especially DHA, and cognitive function, visual acuity, and overall brain development. In the human infant, brain development undergoes its most rapid and complex growth during the last trimester of pregnancy and the first 2 years after birth. This means that during this period, the child's eventual neurological development is highly dependent on its dietary intake of essential nutrients, especially omega-3 oils. Because the fetal and infant brain is unable to convert enough alpha-linoleic acid, an omega-3 oil found in some plants, into DHA, the baby almost totally depends on its mother for its supply. There's good evidence that the placenta selectively takes up DHA so as to ensure an adequate supply for the growing baby. Some 70% of the energy supply during fetal development is devoted to brain development, and lipids make up 50 to 60% of the structure of the brain, with DHA making up 30% of the brain and 50% of the retina's structure. However, since the baby depends on the mother's supply of DHA, omega-3 deficiencies in the mother can lead to increases in DHA deficiency in the infant's brain with each successive birth. Studies have shown that brain DHA deficiency is limited to about 30% with the first generation, but by the third generation, brain DHA levels can fall by as much as 85%. The average DHA and EPA intake combined in the US is about 100 to 200 milligrams per day, far below the 650 milligrams of DHA plus EPA that experts recommend for healthy individuals. Furthermore, it's been shown after birth, DHA levels fall between the age of 6 and 12 months, mostly due to low DHA content in most baby food and formulas. And this can have a profound effect on postnatal brain development. A study that measured the impact of DHA on visual development in 6-month-old infants found retinal and the visual cortex maturation was greatly improved in infants who were receiving DHA-enriched egg yolk versus the control infants who were deficient in DHA. In fact, the researchers found that even in breastfed infants, DHA levels fell, whereas levels rose by 34% in the supplemented infants. In developing babies, higher levels of DHA are needed for the growth of neuronal cells than other brain cells. The composition of the membrane of neural cells is under constant reconstruction, and it can change drastically with changes in diet. It's been shown that while other fatty acids plateau at birth, DHA levels actually double, mainly during periods of intense nerve cell development, that is, during intense brain growth. Overwhelming evidence for the benefits of DHA comes from a recent review of some 50 studies, which concluded that higher DHA in babies' diets translates into better brain function, especially for cognitive and visual function. Further evidence that DHA may promote healthy nervous system development comes from a recent study in which researchers divided 53 normal, healthy infants into those exclusively breastfed and those exclusively bottle-fed with a formula containing no DHA. Using assessments of visual, auditory, and sensory perception, they found abnormalities in all 3 tests in the bottle-fed babies at 1 year of age. A devastating consequence of low DHA status is its impact on the development of neurological abnormalities. In another study, researchers measured DHA, essential fatty acids, trans fatty acids, and arachidonic acid levels in the umbilical artery and vein of infants, and they found that infants with neurological abnormalities at birth had significantly lower levels of arachidonic acid and DHA and higher trans fatty acid levels. In contrast, infants with higher arachidonic acid, DHA, and essential fatty acid levels had more normal neurological function. In a further test of cognitive development in infancy, researchers found that maternal and fetal DHA levels measured at birth correlated with attention and distractibility at 12 and 18 months, with those having higher DHA levels faring better than those with the lower levels. It's also interesting to note that DHA plays a major role in muscle function and coordination in developing infants. A recent study using newborn baboons revealed the highest levels of DHA are found in the motor control areas of the brain, and this also plays a major role in memory, social development, and behavior. So we see that adequate DHA in the mother's diet is critical for the normal brain development in her babies, and that with the first baby, her DHA levels will fall even more because the placenta will preferentially extract the mother's DHA for the baby. This means that subsequent babies will have even lower DHA levels as the mother's DHA is further depleted. The big question is, can developmental problems triggered by low DHA levels be reversed? The evidence seems to indicate yes. For example, in one study, rats who were fed low DHA diets for 3 generations from birth, producing severe deficiencies in the brain DHA levels, and then fed a DHA-enriched diet for 6 weeks, achieved the same level on testing of spatial learning as the animals who maintained an omega-3 adequate diet for the 3 generations. A DHA-enriched diet for less than 6 weeks had no effect. However, the rate of recovery of brain DHA varies among studies, depending on the experimental animal model and other variables. In another study, rat pups that were fed DHA-depleted diets for 2 generations, followed by an omega-3 containing diet, beginning at 15 days after birth, began to regain brain DHA, essential for the brain structure, within 1 day, but the full recovery took 1 month. This slow recovery in rats was confirmed by other scientists. In the rhesus monkey, recovery of brain DHA can take as long as 12 weeks. In addition, the brain recovers its lost DHA slower than the peripheral tissue. In one study, the rate of recovery of brain DHA went from 19% at 1 week to 35% at 2 weeks, up to 80% at 8 weeks. In the retina, which has an even higher DHA content than the brain, recovery was faster than the brain, reaching 72% at 4 weeks, and was fully recovered at 8 weeks. Recovery of DHA by the adult brain, however, is slower and less complete than the infant brain. While DHA is essential for the proper development of the infant brain, it also plays a vital role in the ongoing structure and function of the adult brain. It's a component of several important phospholipids, with the highest level of DHA being found in phosphatidylethylolamine, then phosphatidylserine, and the lowest levels in phosphatidylcholine. Structurally, DHA comprises 22 carbons and 6 double bonds, making it the most unsaturated fatty acid in cell membranes and an important ingredient in increasing the fluidity of cell membranes. Changing the fluidity of cell membranes alters their physical properties, such as their permeability and their protein activity. This change can drastically alter cell signaling, and it plays a major role in increasing membrane permeability of cancer cells, making them more susceptible to immunologic and chemotherapeutic killing. EPA, however, has much less effect on the membrane fluidity than DHA. It's also been shown that a greater incorporation of DHA into cell membranes results in dramatically less susceptibility to lipid peroxidation and oxidative stress in cells, especially the neurons. Although many view the brain as a stable structure that changes little after adolescence, it is in fact always in a state of flux, not only by forming millions of new nerve connections, but also by replacing and altering its biochemical makeup, especially membrane lipids. Connected with this constant turnover of brain lipids are the phospholipase enzymes, and these can release arachidonic acid and DHA from the cell membrane. Dietary changes can therefore drastically alter the brain lipids, which can significantly alter brain function, even worsening neuropsychiatric disorder. Furthermore, there's evidence that as we age, the distribution of DHA in the brain changes. For example, during infancy, the highest level is found in the striatum, associated with motor control, and there's lower levels in the hippocampus, linking the nervous and the endocrine system, and the hippocampus associated with memory. In adults, the highest levels are in the cortex, essential for cognition, and the lowest in the medulla, which is crucial for autonomic function. With aging, the highest levels are in the cortex and the cerebellum, involved in motor control. An example of this region specifically was seen in a recent study when scientists restored dietary DHA to rats who'd been deprived of the nutrient. They found that all areas of the brain's DHA were restored after 12 weeks, except for the medulla, which recovered only 62% of its DHA. These findings therefore provide plentiful evidence that consuming enough DHA may be essential for the upkeep of the adult brain. In the past, a lot of attention has been paid to the effects of omega-3 oils on membrane properties, which are very important for the brain's healthy function. After all, membranes regulate entry into the cell, as well as control receptor function, and this facilitates cellular communication. More recently, however, researchers have discovered that omega-3 oils, especially the DHA component, also affect cell signaling. Hundreds, if not thousands, of special molecules are involved in cell signaling, and these control not only the cell's internal communication with the outside, but also the communication within the cell. A group of special transcription molecules allows cells to communicate with the genes also. The vast array of cell signaling molecules allows the signals to be fine-tuned and alternatively switched to an assortment of functions, such as regulating cellular energy via the mitochondria, activating or silencing genes, generating special proteins, ion regulation, and regulating inflammatory mediators. The complexity of activations within a cell is therefore carefully regulated by cell signaling molecules. Numerous enzymes such as adenyl cyclase and protein kinase A involved in energy regulation are upregulated by DHA. The DHA omega-3 also reduces pro-inflammatory mediators like prostaglandin E2, thromboxanes, and leukotrienes, and increases the production of anti-inflammatory compounds such as lipoxins and resolvin, along with the substances that protect brain cells called neuroprotectins. DHA also maintains the activity of a key cell membrane enzyme called Na+K+ ATPase, and this extracts energy from the ATP to drive the cellular sodium pump, and this controls electrical impulses between the cells. For example, it's been shown that feeding rats a DHA-deficient diet lowers this enzyme's activity. This enzyme is also linked to 60% of the energy consumed by the brain. Maintaining cellular energy levels is particularly important in preventing excitotoxicity, a pathological process that damages or kills nerve cells, since low energy can dramatically increase sensitivity to this destructive process. Another essential cognitive process controlled by DHA is regulating the brain's concentration of phosphatidylserine, which is also vital for cell survival. Diets low in DHA reduce brain phosphatidylserine, and this affects cell signaling for survival through enzymes including the Na+, K+ ATPase, and the calcium uptake. Calcium is one of the most common and versatile cell signaling systems. DHA, but not EPA, has been shown to be a major regulator of the calcium oscillations, the waves within the cells, which regulate a vast array of cellular functions, including neurotransmitter release, mitochondrial function, gene activation, oxidative stress, and, in the developing brain, a neuron migration and maturation. Low DHA levels are also known to lower brain and cellular growth factors, such as brain-derived growth factor. Another detrimental effect of low DHA is to increase neuronal death. An observation of this was seen in a lab study, which revealed a dramatic increase in DNA fragmentation of DHA-depleted neurons in cell culture. Enriching this cellular medium with DHA protected the neurons, an effect that appeared to operate through phosphatidylserine via its actions on cell signaling survival pathways. As well as killing neurons, diets low in DHA decrease phosphatidylserine levels in the hippocampus region of the brain, associated with memory consolidation, and this can explain in part the improvement in cognition and memory that's seen in studies of phosphatidylserine supplementation in patients with dementia. DHA is the major omega-3 oil component of the central nervous system, and it accumulates particularly in phosphatidylserine. Omega-3 deficiency has been correlated with approximately 30 to 35% reduction in phosphatidylserine levels in key neuronal cells during both prenatal and postnatal periods. One of the principal cell signaling pathways affected by both EPA and DHA is the peroxisome proliferator activated receptors, or the PPARs. When activated, this pathway inhibits pro-inflammatory cytokine production, thus greatly reducing inflammation, both systemically and in the brain. And this could play a major role in reducing the damage generated by a host of disorders like diabetes, atherosclerosis, autoimmune disease, brain aging, autism, and neurodegenerative disorders. Studies have shown that EPA and DHA activate the PPARs, which in turn suppress nuclear factor kappa B, the main transcription system that activates inflammation. Thus, DHA, which is more potent than EPA in activating the PPARs, might have important implications for reducing inflammation in the brain. A high intake of DHA and EPA also reduces inflammation by displacing arachidonic acid, a pro-inflammatory precursor, and cholesterol from the cell membrane, reducing the starting material used to make inflammatory mediators. Furthermore, DHA inhibits inflammatory eicosanoid production induced by the release of arachidonic acid from the cell membranes. As we age, brain inflammation progressively increases, but a diet higher in DHA relative to other fatty acids might reduce this age-related brain inflammation. The beneficial effects of DHA extend beyond promoting the optimal neural function and reducing inflammation. A number of studies have shown that omega-3 oils, especially DHA, can also support healthy levels of neurotransmitters, the chemical messengers between the cells. For example, it's been shown that a DHA supplemented diet can restore neurotransmitter release in the hippocampus and can reverse age-related impairment of memory acquisition. This might be due in part to DHA's effect on increasing the fluidity of the cell membranes, which improves their ability to release the neurotransmitters. But it might also involve cell signaling. Another important effect of DHA is increasing the density of the nerve outgrowth called the dendrites, a property that's strongly connected to improve learning in rats as well as humans. In addition, DHA has a rather complex effect on a number of neurotransmitters, including serotonin, norepinephrine, acetylcholine, glutamate, and dopamine. Deficiencies in DHA have been shown to lower the levels of dopamine and the dopamine receptor D2 in the frontal lobe, which can result in problems with attention and learning. This can explain the ability of DHA to promote focused attention and to calm anxiety. One important effect of a DHA deficient diet was seen in a study using the reward chemical dopamine, and it found that long-term omega-3 deficiencies raised dopamine levels in an area of the brain closely linked to addictive behavior. This, in essence, significantly increased the susceptibility to addictive behaviors, whether to foods, sex, or illicit drugs. Finally, DHA also plays a role in neurotransmitter receptor function. Recent studies have found significant effects of DHA on cell membrane structures called G-protein coupled receptors, which communicate with the inside of the cell when stimulated by neurotransmitters such as glutamate, serotonin, and acetylcholine. Since these receptors are inducible, that is, their numbers can be increased on demand or by altering nutrients, dietary changes can significantly affect how the brain functions. Because of the frequent profound effect of EPA, and especially DHA, on cell signaling, membrane neurotransmitters and receptors, one would expect that omega-3s produce equally profound changes in human behavior. And in fact, a number of studies have found a close correlation between omega-3s in the diet and behavior. Of special interest is the effect of omega-3 oils on depression, suicide risk, aggression, anger, and anxiety. Depression has become a major problem, with rates increasing at younger ages than ever before, and the disease occurring even in pre-teens. Also increasing are the rates of anxiety disorders, anger discontrol, insomnia, violent suicide, and even homicide. Scientists have proposed a common link between these disorders and chronic brain inflammation. Likewise, a number of medical conditions have been associated with chronic depression, like cardiovascular disease, cancer, and overall poor health. Until recently, it was assumed that this was because of the neuroendocrine effect or other non-specific reasons. However, a growing amount of research now indicates these diseases are linked to depression by a common mechanism, a high level of inflammatory mediators such as cytokines and inflammatory prostaglandins. Unlike acute brain inflammation as seen with meningitis and encephalitis, subtle chronic inflammation can be silent except for its behavioral manifestations. Chronic brain inflammation can be the result of a number of conditions, such as abdominal obesity, latent viral infections, autoimmune disease, metabolic syndrome, heavy metal toxicity, and deficiencies in omega-3. Studies have revealed a correlation between chronic inflammation and depression. A recent study found a strong correlation between the inflammatory cytokine levels and the high rates of depression and suicide. Furthermore, a number of studies suggest that many of the antidepressant medications also act as anti-inflammatory agents, and that patients who respond poorly have the highest levels of inflammatory cytokines. Chronic brain inflammation has been found in post-traumatic stress disorder and postpartum depression. It might be the omega-3s improve major depression, as well as a number of other neuropsychiatric disorders by reducing the brain inflammation. It's known that a deficiency in omega-3 oils affects serotonergic and dopaminergic neurotransmission, and if restored before weaning, these deficits can be repaired in the brain. Both of these neurotransmitters, as well as inflammation, play a significant role in neuropsychiatric disorders like schizophrenia, borderline psychotic personality, suicide, hostility, aggression, bipolar disorder, and major depression. Shrinkage of the hippocampus of the brain has been described in major depression and in post-traumatic stress disorder. This shrinkage or atrophy is also accompanied by deficiencies in recent memory. Memory deficits may occur before atrophy and neuron loss, and treatment of the depression may help to restore hippocampal neurons and connections, the neurites and the synapses. While both EPA and DHA have been associated with improvements in major depression, only DHA stimulates neurite outgrowth and synaptic development and repair, that is, brain plasticity. Therefore, it might be a better choice in methods to repair the brain damage of chronic depression. A growing problem in the country is postpartum depression, which is often linked with suicide, homicide, and infanticide. A number of studies have linked low blood levels and red blood cell levels of omega-3 fatty acids with postpartum depression. For example, a study of 10 pregnant women who developed postpartum depression versus 38 who did not found the proportion of DHA in the serum phospholipids was consistently lower in the women with the postpartum depression. Suicide risk has also been linked to low omega-3 levels in the blood and the cell membranes. In one study, researchers at the Department of Neuroscience at the New York State Psychiatric Institute followed 33 medication-free depressed patients for 2 years. 7 of the subjects attempted suicide. Low plasma DHA percentage in serum phospholipids and a high ratio of omega-6 fats to omega-3s accurately predicted the suicide attempts. Unfortunately, Americans consume far more omega-6 than is needed for good health. With the aforementioned effects of DHA on the brain levels of neurotransmitters serotonin and dopamine, as well as receptor expression and cell signaling in synaptic membranes, one should not be surprised by the dramatic behavioral impact of an omega-3 deficiency. The evidence for the effects of omega-3 oils on another neurotransmitter, glutamate, the most abundant messenger in the brain, is still being debated. There's compelling evidence that a number of neuropsychiatric disorders like schizophrenia, bipolar, suicide risk, and major depression are related to abnormalities in glutamate neurotransmission. Data to indicate that excess glutamate might be responsible for major depression, as well as other psychiatric disorders of mood and impulse control, comes from a number of quarters. Firstly, depression and schizophrenia have been linked with a decrease in glutamate transport proteins, which normally carry excess glutamate away from the nerve endings. This can greatly increase brain glutamate levels and excitotoxicity, and would explain the loss of neurons in the hippocampus seen in schizophrenia and major depression. Secondly, the drug ketamine, which is a powerful glutamate receptor antagonist, also acts like an antidepressant. Another drug, riluzole, which prevents the release of glutamate from the cells, also prevents degeneration of brain cells in depression, and it raises brain growth factor levels, which are consistently lower in depression. Finally, careful studies have shown that glutamate receptors in depression are overactivated, and glutamate removal systems, which normally protect the brain from glutamate excess, function abnormally. These same studies have found that the GABA system, which is essential for protecting the brain against excitotoxicity, also works subnormally. If indeed major depression, bipolar disorder, schizophrenia, and suicide risk are related to excitotoxic injury, omega-3 oils can play a significant role in combating these disabling disorders, since several studies have shown omega-3 oils, especially DHA, significantly reduce excitotoxicity. Low omega-3 levels, especially DHA, have also been associated with anger and hostility reactions. For example, in the first of a 2-part multi-center randomized study, medical students were given either a placebo or fish oil containing 1.5 grams per day of DHA for 3 months. In part of the study, which was done during high stress periods during the final exams, they found an 8.9% increase in aggression in the placebo group, whereas those who were taking the DHA experienced no increase in aggressive tendencies. In the second part of the study, using 46 non-stress students, DHA had no effect on aggression. This is because increases in aggression were secondary to the stress, and DHA appeared to only calm stimulated aggressive urges. This is consistent with the above-cited studies indicating DHA deficiencies cause disruptions of monoamine neurotransmission, dopamine, norepinephrine, serotonin, as well as acetylcholine and glutamate transmission, all of which are related to impulse and anger control. These effects are also related to cell signaling, anti-inflammatory, and membrane fluidity effects of DHA, which are greater than those of EPA. In a later study in 41 university students, researchers compared a high DHA diet 1.5 grams per day with a placebo over 3 months to see the effect on hostile responses. They found there was a 27% incidence of hostile answers at the baseline, which rose to 92% during the final exams. Again, there was no increase in hostility among those who were taking the high DHA supplement. They also found DHA supplementation significantly reduced hostility measures among the university staff. There's even evidence that extra DHA in the diet can reduce aggressive behaviour in younger children. For example, in a study examining girls and boys aged 9 to 12, adding omega-3 fortified foods, 3,600 mg DHA and 840 mg EPA per week reduced impulsive behaviour in both males and females, with stronger results seen in females. One of the effects of higher DHA intake is to reduce plasma norepinephrine levels, and this reduces the fight or flight response. In one study, DHA's ability to modulate plasma norepinephrine was quite dramatic, with no effects on cortisol, epinephrine, glucose or insulin levels. DHA could thus be a useful weapon to reduce anxiety and panic attacks. Healthy adults should consume 240 to 1000 mg of DHA daily to protect their cognitive and neurological health. While many adults already consume omega-3 oil supplements containing both DHA and EPA, DHA standalone products allow those seeking higher DHA-EPA ratios to customize their intake according to their unique health concerns. Pregnant and lactating women require a minimum of 300 mg of DHA daily to promote optimal nervous system development in their children. Parents should ensure that their young children consume at least 150 mg of DHA per day to support health. Omega-3 oils are also available in good-tasting liquid formulas for young children and those who have difficulty swallowing capsules. From this review, we've seen omega-3 oil plays a major role in brain development, with the highest demand occurring during intrauterine development and the first 2 years after birth, during the brain growth spurt. Deficiencies during neurodevelopment in the mother alter not only immediate development, but can set the stage for later disease and brain malfunction. Because of the high demand for DHA by the developing baby and the preferential extraction from the mother's supply by the fetus, deficiencies are compounded with succeeding births should the diet not improve. Likewise, this has a profound effect on the psychological profile of the mother, with severe deficiencies of her DHA resulting in increased rates of postpartum depression and concomitant increase in the risk of suicide and homicide. We've also seen that the effects of DHA and EPA on the nervous system go beyond the structural membranes or the fluidity, but they rather influence the cell signaling, the neurotransmitters, and the receptor expression and function. This also includes gene regulation and expression. The effects of omega-3 oils on inflammation are complex and go far beyond physically disabling arachidonic acid, the starting material for inflammatory prostaglandins from the cell membranes. DHA also increases lipoxins, inhibits NF-kappa B, and produces neuroprotectin D1 and resolvins. Because of the complex interaction of omega-3 oils with a magnitude of systems, both cellular and via neural networks, variations in brain EPA and especially DHA can have profound effects on learning and behaviour. Ensuring optimal amounts of these healthy oils is essential throughout the healthy lifespan. For more information on herbs, supplements and natural treatment plans, check out my website.